Well, hello there. This is your favorite online biological science laboratory instructor trying to get us all back on track. All right, this is going to be exercise 10. We'll be fine, okay? Exercise 9, I've already posted a video coverage of it. And for this one, it'll be this video, and then you will have some things to do, okay? One that I did to help is I changed the instruction sheet. Don't worry about that handwritten 90 and 91. I've made that change. But I'm going to go over that, okay? So it's called cell division, mitosis, and meiosis. So these are two cell division processes that are very, very important. That's how you got to be in existence, and that's how you have grown to the size that you are, and you repair and you do what you do. So they're very important processes. So let me just go over the instruction sheet, and that'll help you, okay? It's exercise 10. So on page 103, I'll just read it. It says, review the learning objectives and instructions on page 103, paying attention to the terms that are in bold print. And you know that from textbooks in general. And the bold print terms are mitosis, cytokinesis, cell cycle, G2, G1, and we'll look at that, okay? And then carefully review the cell cycle diagram shown in figure 10.1, and it's on page 104. So look at the diagram or look at your lab book and look at page 104. And notice it says the cell cycle. Cell cycle, a cycle is, it starts here, it goes around, and it does it again, okay? Not all cells in our body do the cell cycle, at least not the same way. But when they do, it starts and the cycle has different parts. G1, S, G2, and then mitosis where the cell divides. So I'm not going to go into all that because in your lecture you've had the cell cycle. But the cell cycle is so important. And you want to know, I mean, you being the bigger you of humans, is what controls the cell cycle. Why is it that in your body, okay, you have certain cells that go through the cell cycle quickly, a little bit slower, some that don't go through it, and what stimulates them to go through it. And when they go through the cell cycle, what could come in and cause a problem, and they go through it too fast, and that's how you get cancer, you get tumors, okay? So it says carefully review the cell cycle. And then I have a video link provided that says watch the cell cycle video. So I provide a link for that. It's right there, okay? And then number 3, review the mitosis stage description and diagrams shown on page 104 and 105. And that's in your lab book. So it has mitosis description, and then it has diagrams on page 105. All right, and then also I've included in the packet some diagrams right here to go through it. So what you could do is label, for your own purposes. I've done, several. So what you can do is go through and label those to reinforce your learning of the different aspects of mitosis. And I have a video on mitosis, okay? And I say here, shown below are diagrams for animal cell mitosis. There's animal cell and plant cell. Now, we're animals, but what you typically use to manipulate mitosis is you use plant. And the plant that's used is onion. Scientific name for onion is allium. Okay? So at the bottom of my instruction sheet, I have compare the onion and whitefish micrographs on page 106. So in your lab manual, turn to page 106, and you'll see light micrograph. A micrograph is a photograph taken through a microscope, okay? And at the top it shows plant, and at the bottom it shows animals. Plants, by and large, are rectangular and boxed and hard, and animal cells are more roundish, okay? And where you can really tell a difference is look at diagrams D for 10.3 and 10.4. Look at D. Notice in the plant, the one at 10.3, there's a line that goes across called the cell plate. And that's going to be a precursor of the cell wall to divide the cell. And in the whitefish, in D, it pinches, like you take some dough or something and pull it apart, because it has a membrane that can allow that to happen. And that's how cytokinesis occurs. Mitosis is made up of dividing the chromosomes, karyokinesis, and then the cytoplasm, which is cytokinesis. All right, then in the instructions I have number 5, read through the meiosis descriptions and carefully go over the diagram in figures 10.5 and 10.6 on page 107. So look on page 107 and put 100% of your effort into mitosis and 70% into understanding meiosis. Meiosis and meiosis, excuse me, sneeze, okay? Mitosis is to give you identical cells. I say it's like a photocopy machine, and meiosis is to cut your chromosome number in half, and I call it like shuffling the cards and then dealing them out. You have a whole deck of cards, and you shuffle them, and then you put card here, card there. And that's like the chromosomes in you, or in a male, female, are all the chromosomes, but what you put in the egg and sperm is 1/2 of the number. So there needs to be a way to cut that chromosome number in half. We have 46 chromosomes, but what shows up in the egg and the sperm is half that number. It's called haploid. The total number is called diploid, okay? And it's a process to give you haploid cells, and there are 2 divisional processes. And as you look at the diagram on page 107, carefully go through it, you can see it. And then on page 108 is a comparison. It says summary of mitosis and meiosis, where they take the diagrams and go through, and you can see one does a division process once and the other one does it twice, okay? So anyway. Okay, and then for the material and procedures for examining the slides, look at the video on it. So there's a video on how to prepare onion root tip slides. Okay, you're not going to do it, but you can watch it, and there's a video to show you the whole process. You're going to, omit pages 113 through 116 and 118 through 123. So you're not going to have to do anything that's on pages 113 and 116 and 118 and 123. Now, the one in between is page 117, and that's where the post-lab questions are. So you're going to do those and submit those, and you'll submit the pre-lab questions that are on page 111. All right, so now, here's an activity that's not in the book. I mean, it's kind of in the book, but it's not put forth like I have it. It's called Calculating the Mitotic Index, and there it is there. And what you're looking at is how many cells are in some stage of mitosis and how many cells are, compared to all cells. It has the instructions of how you're going to do it. So you're going to do 2 things. You're going to do the mitotic index, and then you're going to break down and see how many cells are in some phase of mitosis. Okay, so let me go back. Activity 1 in the instructions will have you go through, and what you would do is be counting. You'll just count. Just start here, count down, go up, over, down, up, down, over, until you've counted 100, 200 cells. And I have 1 micrograph. I have another micrograph. I have 3 micrographs for you to use, okay? And what you do is go through it first, and is it in mitosis or not? Calculate that until you've done 200. And let's say you have 56 that are in some phase of mitosis out of 100. So that's 56%. 0.56 will be the mitotic index. You divide the total by the number of cells. And then of the cells that you have, you want to see how many are in prophase, metaphase, anaphase, or telophase only. And what that does is it gives you some idea of the duration of those phases. So mitosis, say, lasts 90 minutes, okay? So you look at the cells in some phase of mitosis, pro, meta, ana, tele, and depending upon the percentage, you multiply that times the 90, and it tells you how long each phase lasts, okay? So there's a question about that. On the back page of the cheat sheet, the instructions are 2 animations for meiosis to kind of help you get a better understanding of it. But the main feature is mitosis, okay? And you don't have a lot to turn in, all right? And that's it. And you do the pre and post questions. And you don't have to do anything, because those are the pages I said to omit. If you had been, like it says, show pictures of mitosis and all that, you would just go to Google, put in meiosis, metaphase 1, and look at the micrograph. But you don't have to do that, okay? That's all right. You don't have to do it. I think we've given you enough. All right, so I hope that this helps on my 10-minute closure here. And this is exercise 10. And then after this, we'll be straight for the rest of the semester. All right. I hope that this helps. All right, and we'll post this and get it going, and it's all going to be good. Okay, and I'll post the quiz on it. All right. Goodbye.